So this is week 6 of Micro 2, and what I've drawn on the board is just a brief schematic of what we've talked about so far this week. Basically where we're starting is right here with the end of the intestinal nematodes. Up until the midterm, we've done these. So I'm going to start with these, and next week we're going to do the second group of the flatworms. Okay? So starting at the top with the intestinal nematodes, we have our strong woman, as I like to call her. She's an intestinal roundworm that is parthenogenic, which means that she's able to reproduce sexually on her own. She makes her own gametes, and she doesn't need the help of a male to reproduce. She's also capable of auto-reinfection, which means that her eggs are going to hatch inside of the intestines of the host. And so basically, the host is constantly reinfected over time because the eggs aren't released out of the body and reconsumed in order for infection to occur. These are the main points from this intestinal roundworm. Everything else is available through notes, and it's roughly similar to hookworms, so I'm not going to go over everything. The next intestinal roundworm is Anissa. This is my other friend over here, Anissa. She's also called the herring worm. So you can just remember that because her last name is Marina, as in marine. Herring are a type of fish, so marine herring worm, okay? Small fish are her intermediate host, and birds and marine animals are her final host. So humans are also a type of intermediate host, but they're not the main host involved in this roundworm. In this roundworm, the larvae are the infective stage, and that's pretty much, again, the gist of what's going on with this worm. The rest is available through notes. Okay? So now we get through the tissue nematodes, and we have 3 types. We have the kind that invade lymphatics, the kind that invade subcutaneous tissues, and then we have this one kind of offset. It's not really a filarial, but it's still a tissue roundworm. Okay? The lymphatic nematodes are listed here, and the main difference that she uses to differentiate them is just their vector. The first one is infected by the cold Anopheles mosquito, and the second one is the second vector is the Monsonia Anopheles mosquito. For our subcutaneous tissue roundworms, we have Loa loa, which is also called the eyeworm, and we have Anchocerca, something like that. And she also is called the blinding filaria. So that's another word for this one. The vector for Loa loa is the chrysops fly, and the vector for the Anchocerca is the simian fly, also called the black fly. The life cycle for all 4 of these are roughly similar. They're listed in the notes again, but the main thing to understand about these life cycles is that the filarial form is the adult form, the microfilarial form is the larval form. The larval form is the infected form. These are also transmitted by way of insect bite, and they're not directly consumed. So that's another thing to remember about these 4. Now the one that we kind of have offset to the side, again, it's not a filarial form, but it is a tissue roundworm, so it's kind of an exception to the rule. This is also called the guinea worm. I wanted to make sure I said that. The common name is the guinea worm, and some of the things that kind of differentiate it from the other ones are that you ingest this directly. So it's not by way of an insect bite, it's directly ingested. We have the larval form as the infective form, as well as the infective form is also in the 3rd stage of development. So if you consume this within the 1st stage of its larval form, you won't get any type of infection. But if it's in the 3rd stage, that's when you actually do get an infection. Okay. The rest is available through notes. The second group that we talked about is platyhelminthes. These are the flatworms. And we have 2 groups, the second one of which we'll talk about next week. Cestota is the group that we focused on this week. So we have Taenia saginata, Taenia solium, we have Heminolepis nana, and Diphyllobortherium latum. Okay? And, these are segmented. So she emphasized that in class a lot. So we have to remember that these are segmented. These are the most extreme flatworms that we have to worry about. So they are the most infective. They are long and ribbon-like. They can be up to 20 meters long, and most of them have to remove by way of manual removal from skinning. Okay. The adult form of these are in the intestines of humans, who are the final host, and the larval form are found in the tissue of intermediate hosts. So intestines versus tissue. I drew a little picture down here. Hope you like my picture of the cestota worms. This is just very general. We have a scolex, which is the head. We have hooklets, which allow the worms to hook onto the intestines of the final host. And then we have the proglottis, and this is describing the entire portion below the neck. Excuse me, that's wrong, I'm sorry. Strobila is the entire portion below the neck. The proglottis are the individual segmented portions, and these contain both reproductive organs, okay? So basically in our life cycle, you'll see that this portion of the proglottis will kind of fall off, and it'll mature and the eggs will hatch in the environment as I have listed here. So the end of it will fall off in the environment, the eggs will hatch, and that's part of our life cycle that you can see in the notes. Here I kind of drew a spectrum. The newer reproductive organs are towards the head, and the older ones, or the more mature ones, are towards the tail. And so that's why the tail is going to fall off and produce the hatched eggs. Taenia saginata, as we have here, this is the beef tapeworm, and Taenia solium is the pork tapeworm. It's important to differentiate between those two, so definitely look in the notes for that. But Taenia saginata is definitely larger. It contains up to 2,000 protoglottis, so 2,000 of these segments. They have the scolex with suckers and not hooklets. However, Taenia solium have the scolex with hooklets and suckers. Okay, so that's another differentiation. Let's see, Taenia solium, it's found in pork, undercooked pork. They are smaller. The infective stage is in two forms, in the egg stage and the cysteria stage. The human is the intermediate host in this situation in some cases, but for both of them, humans are most of the time the final host. So that's important to remember. Final host, here we have final and intermediate. Okay. Life cycle is found in the notes, and we talked briefly about these two this week. Not much of a differentiation with those, except for this one is the most common in the U.S. So that's probably something that might show up on a test as a multiple choice question. That one is also found by way of feces and mice. So if it's consumed by humans by way of food or whatnot, that will be infective to humans. And the last one is the broadfish tapeworm. That's the common name. The first intermediate host is the copepod fish, and the second intermediate host is freshwater fish. Humans are the final host. Life cycle is found online, and some of the symptoms that I just kind of thought kind of stood out were the intestinal blockages that are caused by this, and as well as a nervous system disorder due to vitamin B12 deficiency. So this is just a brief overview of what we talked about week 6 in Micro 2. Have a good day.